Okay, good afternoon, everyone. We will start where we left in the last class. So we were discussing about repair mechanisms, and in that we have learned about replication errors. In that we learned about how DNA polymerase could make mistakes, and it may add up or incorporate a wrong base that is called as mismatch. And we also have seen about we have discussed about mismatch repair mechanisms, right? And we also have seen the importance of methyl groups, and we have learned about what would happen if exonuclease activity should go this way, as opposed to if exonuclease activity should go this way. To be precise, should it be, if it were 5' to 3' exonuclease activity, how is it solved? And if it is 3' to 5' exonuclease activity, what kind of exonuclease is utilized in that situation? So I also told you to read about the eukaryotic mismatch system. I hope you have done, you have read it. If not, please go to the textbook and read it. I also told you to make a note of different diseases associated with mutations in repair mechanisms. So as we kept, we have also tried to learn about spontaneous mutations like hydrolysis, deamination of the nitrogen bases or the base itself. And what we are going to learn is about other types of mutagens. You can have chemical mutagens, you can have physical mutagens like, say, radiation and so on. So we'll read quite a few of them. And before going there, we will have where we left, that is AMIS test. AMIS test is, the objective of AMIS test is to evaluate or estimate the mutagenicity. Okay, that is the objective of AMIS test. So I hope you know what is a mutagen, a compound that could cause a mutation. A mutagenicity is the degree of mutation. How many mutations can a compound do? The potency of the mutation. Carcinogenicity is the ability to cause cancer. Cancer is caused by mutations. All mutations don't lead to carcinogens or cancers. Okay, some do and some don't. Well, they are related in the sense that all carcinogenicity is mutagenicity. Not all mutations is carcinogenesis. And we also described about prototrop, that is the wild type, the organism naturally found in natural environments. That means it should have a lot more capacity than those that are grown in the lab in general. Auxotrops are mutants. That means they have a mutation in one or more of metabolic genes. They are very useful tools for studying in molecular genetics. Quite a lot of things were done using auxotrops. And that is the reason why I'm also spending time trying to describe the use of AMIS test. And that gives us an opportunity to learn some things about how molecular genetics is studied using bacteria. And then we also learned about minimal medium. Minimal media is something in which the components or nutrition, CHNOP, something like this, they are given as inorganic compounds, majority of them. So the bacterium should be able to utilize those materials and make their own components such as amino acids, vitamins, and everything else from the nucleic acids and so on. Whereas that is opposed to rich medium, in which when nutrient agar, for example, nutrient broth, we give yeast extract and so on. That means it has a lot of peptides and amino acids, proteins, which are taken up, digested, to amino acids and used up radially. And spontaneous mutations that we have seen, spontaneous mutations happen. That means they occur naturally. So we take a bacterium, okay, we'll call this a bacterium Salmonella, say, and it is His minus. Yesterday I told you what do you mean by His minus. It means, say this is the bacterium, okay, Salmonella, and it is His minus. That means it cannot synthesize histidine by itself. So it is an auxotrope. What would the prototrope be? The prototrope will be able to synthesize its own histidine from other from basic components. Whereas in this particular strain, the Salmonella is His minus. That means it is deficient in producing required to synthesize histidine. Okay, histidine is an amino acid synthesized by several enzymes from scratch to, or convert from other molecules to histidine. So if you, I will give you a comparison. I'll try to use this space. So let's do it this way. So I have Salmonella, which is wild type. Okay, I'm just putting it like that for ease of understanding. And I will also put here the other one is Salmonella His minus. Okay, that means this is the first one. Wild type I'm saying is the prototrope, whereas His minus is the auxotrope. Okay, so some spontaneous mutation happened because of which this particular strain has become His minus. So now we already discussed about rich medium and minimal medium. If you give rich medium, wild type or the prototrope will be able to grow. If you give rich medium to this strain, the auxotrope, there also it will grow. Why does it grow? Because rich medium has already, we are supplying the bacterium with ready-made histidine. So it will just directly take, you make use of it and grow. It doesn't have to synthesize histidine, right? Now what will happen if we grow it in minimal medium? This is a wild type strain. That means it has all the genes required to synthesize what it needs. So it will grow. Now take minimal medium here. This particular strain, say His minus strain, the auxotrope, cannot grow on minimal medium. Why cannot it grow? So if it has to grow, it needs a lot of proteins. And majority of the proteins will require the presence of histidine. Histidine is a charged molecule. Maybe it has some important functions in interacting with other molecules, or maybe it's in the active site of some of the enzymes. So if there is no histidine, then the bacterium cannot grow. So that is why if there is no supply of minimal media, no supply of histidine, then the strain does not grow. Now think of a small adjustment. What will I do? I am minimal medium plus I will give histidine. I have given minimal medium plus histidine. What will happen now? Can anybody answer? I'm also giving more situations plus leucine. Minimal medium plus lysine. What would happen? In all the cases, because the wild type can grow in minimal media, so it will be able to grow in all the other combinations. I want you to tell me what will happen to this strain. That is His minus strain, the auxotrope for histidine. Will it be able to grow in this situation? If histidine is supplied, it will be able to grow. Why is that? Because it is deficient only in histidine. metabolizing or actually synthesizing histidine. So if you supply histidine in addition to minimal medium, then it will be able to grow. If you supply minimal medium plus leucine, it cannot grow because it is still failing to synthesize histidine. That is how people find out or isolate auxotropes. Okay? And the same is the case, it will not grow here also. I hope I gave you some basic idea at least, which is of a bacterial genetics, the base for bacterial genetics. What is minimal medium? What is spontaneous mutation? I'll do something else now. In that, what I'm going to do here is say some mutation has happened. Because of that mutation, okay, his minus, there is a gene, for example, his L, I'm just putting it, or yeah, just say there is a his gene and it has a mutation. Because of that mutation, and it assume that is a point mutation, a substitution mutation. So because of him, this is now his minus. And now what happened is something has happened after several rounds when you grew it in minimal media plus histidine. And what happened was that at the same location, another mutation has happened, because of which it is now functional. Okay? If in the same point a mutation has occurred such that it is now reverted to normal phenotype, okay, that is called as spontaneous revertant, right? It was, what was it originally? Originally, if you see it like in the S wild type, his plus, right? Because a mutation occurred in this place, it became his minus. And there is again a mutation happened in the same place, because of which it became his plus. Right? This is called as spontaneous revertance. Any strain that becomes like that. Okay. Let's get to Amy's test. Amy's test, I told you the function of it, or the objective, is to estimate the mutagenicity of a given compound. So we take Salmonella bacterium, which is requiring histidine, so which should then be, it is his minus. You actually have to write Salmonella strain name full, but for ease, I'm writing like that. So now you take two plates, plate 1 and plate 2, which is filled with minimal medium. Okay? And then when we are making the medium, in this case, we have also added the mutation. And in plate 1, we did not add anything else other than the minimal medium with agar. So after that, we plate the cells on both equal number, 10 to the power 8 here and 10 to the power 8 cells here. Okay? So, and typically what should happen? Nothing should grow, because the strain we have taken is his minus, auxotropic for histidine. But then there are two colonies here. What would that mean? They are spontaneous revertants, the concept I explained to you in the previous slide. Now, in plate 2, where we have added mutagen in the medium, you see more number of colonies. What does that say? If it were just spontaneous revertants, you would find 2 to say 4, right? But you're finding so many of them. That means you can count based on it as, I think, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 18, 19, 20, 21, 22. You have about 22 cells, 22 colonies, okay? Whereas in this one, you only have 2. That means you can say it has produced about 20 more colonies than spontaneous revertants, then that means the suspected mutagen is actually a mutation. For simplicity, I will use the same setup, and I'm just giving data so that you will probably understand better. So plate 1, 2, 3, and 4. Okay, and this is a different experiment except that the setting is same. So in plate 1, I did not add any mutation. In plate 2, I added a mutation called X. In plate 3, I added mutagen Y. And in plate 4, I added mutagen Z. Now, after incubation in the lab at 37 degrees centigrade and so on, I want to see how many colonies have formed. Say, in the plate 1, there were about 4 colonies. 1 second. Yeah. There were 4 colonies in plate 1. In plate 2, I have 10. In plate 3, I have 3 colonies. In plate 4, I have some 40. Now, based on this information, what can you say? In the first plate, there are spontaneous revertants. You did not add anything. That is usually also called as control. In experimental designing, you will learn about, you should know what is meant by a control, and only then you know what is a control. Say, yeah, well, we will discuss that when we get to the lab things. Right now, anything done with a control is an experiment. Anything done without a control is not an experiment. For simplicity, I will also put like this. There is a person, and the person has taken medicine called M1. The person was suffering from fever, and here the person has taken M1 medicine, and the person became well. Okay, is this really medicine or not? Okay, how would you know that? Because this person got well? You can only say whether it is true or false if only you had a control, where there should be a person who has the same disease, did not take the medicine M1, and then you see what happens to this person. If this person who did not take medicine also got well, now is M1 medicine or not? Right? You don't know it. You cannot say anything about it. Maybe it's the immune system of the person who resulted in getting the person better, right? So that is why, in this case, this is the control. Okay? And only you should have something to compare with. Only upon that, and also you will actually need more statistical analysis. You should not take anecdote as evidence. Well, right now we'll just get back to where we are. I hope you understood this important part. That is critical thinking. These are some steps of critical thinking that's useful and important for experimental design. So getting to this, plate 1, 2, 3, and 4. Plate 1 has spontaneous revertants 4. And is X a mutagen? Yes, because at least it is twice that of the spontaneous mutagens. So you can count it as yes. Is Y, is it a mutagen? It is unlikely. It is not a mutagen because it is almost similar to that of the control. So it is not a mutagen. Is Z a mutagen? Yes, it is a mutagen because it is at least 10 times more than the spontaneous revertants. Okay, so you can say that based on this data, X and Z are mutagens, and out of which again you can say Z is more potent mutation compared to X, right? That is what you can do. So we will now have a look at different types of mutagens, and that is how what you should remember is that these kinds of tests are used to figure out the mutagenicity, and that is how they figure it out as well. So first thing, we'll go with radiation. It is relevant to us every day. we have skin color, we are pigmented, we have melanin in our body. It is there for a reason. I'll give you a question, and I want you to find out. You know about Caucasians, that is the Europeans, right? They are almost there is no pigment. Why is their skin very fair or kind of skin colorless? We all originated from Africans, and we are supposed to have a very highly pigmented skin. And in India, we have several variations of skins. And then if you see the Caucasians, the Europeans, they do not have pigment. And I want you to find out why it is so. The logical reason is what we are looking for. Coming to this, what can UV radiation, we are all continuously exposed to UV radiation, and that can cause a lot of problems. Around, we already read about this, that nucleic acids absorb maximum at 260 nanometers. And if you're exposed to UV light for a long duration, we might end up with a cyclobutane ring formation, that is between thymine dimers. You can also call it as thymine dimers. Wherever you find two T's, say A, T, T, G, C, T, and T, T, G. Say, for example, you have a sequence like this. You have two sequences here, right? TT and TT. Not only those, yeah, let me finish this. These two can form a cyclobutane ring that is indicated here. That is a covalent bond formed between these two stacked bases. And it is a kind of damage to the DNA. So they're not only thymine dimers, you would also have thymine cytosine. Wherever you find TC also can form a dimer. It is, these groups get excited because of UV light, and then they interact or form covalent bonds between them. And this is called as cyclobutane ring. Okay? Because of this formation of this thymine dimer, it is incapable of base pairing. So there will be a strand, complementary strand here, sorry. There should be a complementary strand here, to which here it would have been base pairing with, A, and this should have been base pairing with A, and so on. And this would be T. Because of the thymine dimer formation, they cannot base pair properly with this. And also it is a hindrance for DNA replication, even transcription also. Okay? So DNA polymerase, if it is coming to this place and using this as a template, this strand, then it doesn't know what to add. Why is that? Because of the cyclobutane ring, they fail to form proper hydrogen bonds. Okay. So there are other types, chemicals, which are, for example, base analogs. I think I described what it means. Base analogs is, they look structurally very similar to the nitrogenous bases present in the DNA. Here is 5-bromouracil. 5-bromouracil, it is very much similar to that of thymine, right? Uracil, and here if we had CH3, it would have looked like thymine. So somehow it can get incorporated into bases, nucleotides, and nucleotides can containing 5-bromouracil in a it can also exist in keto or enol tautomers. I hope you remember what are tautomers. That is, their composition is same, except that they have a little bit variation in how the groups in the side groups. Yes, in the enol group, enol form, it can base pair with guanine. Normally, if you consider this to be very similar to T, you would expect that it is base pair to adenine. But in enol form, it can base pair with guanine. right? What is the enol form here? I think you have H here, NH, right? And here is a keto group, that is C double bond O here. But in the enol form, what you're seeing here is that the N, it has given away the hydrogen you can think of, or it loses a hydrogen, and the keto group here, which was present on the fourth carbon, is now a OH, hydroxyl group. Okay, hydroxyl group, enol. Keto group is this one. This is the keto form. About whether it is in keto group, then it is a keto tautomer. If it is a OH, then it is a enol tautomer. Yes. So I hope you understood that in enol form, 5-bromouracil can base pair with guanine, and that's a problem. And during replication, it can induce mutations. And I want you to also find out what kind of mutations it will be. Is it transition, insertion, substitution, transversion, all those things? You have to keep all these things in mind. You have to memorize what is each so that you can always determine, or these are some basic things that you need to remember so that you can understand the literature better. There are other enzymes called as intercalating agents. And in that, I told you we will talk about ethidium. Ethidium bromide is what we use in electrophoresis for visualization of DNA. Here it is said as ethidium. And you can also see there are two more, proflavin and acridine orange. There are many others. Some of them can incorporate, can interact with the DNA. Say, for example, I will try to draw a structure, and I hope you can remember what is meant by base stacking, right? Base stacking, yeah, let me try again. So base stacking is, I'm putting as squares or rectangles, so you will still be able to, I hope. These are bases on the DNA, nitrogenous bases, and you can see that they are arranged into spaces like this. This plane and this plane are like a stairs on the ladder. Okay, they are parallel to each other, and that is called as stacking. Okay, that is called as stacking. So if I have to draw the opposite also, the complementary strand, I should use that. Then the opposite strand would be something like this. And they are also stacking one upon the others, right? These are called as stacking interactions, which is kind of, you can think of rings, right? These rings are, if it is A, adenine, it has two rings. If it is A, adenine, and if it is T, you will have one ring, right? If you see these intercalating agents, okay, ethidium, proflavin, or acridine orange, they have these three rings. They also have rings. What does that say? They can slip in between these bases, and they can stack themselves along with them. Okay, this is called intercalation. They are just sliding between. It's like you have a pack of cards and you take another card and put it in between the stack of the cards. It is similar to that these intercalating agents, they just fit in. Why are they fitting in? Because they also have these hydrophobic ring structures, which can interact with these stacking bases like this, as shown here. Okay, that is called as intercalating agent. This is not a covalent bond. It's just hydrophobic interactions. You can think of it like that. They are non-covalent interactions. So if they are present, and then they can cause a lot of problems during replication. They may cause the addition or deletion within the DNA. If a DNA polymerase was reading and it comes across a intercalating agent here, then it may add some base here based on the interactions or the hydrogen bonds that could form. Because of that, additions or deletions can happen. Okay? Let me try if I can explain this any better. Let me try once again. Say, for example, DNA polymerase is polymerizing. So assume this is 5' and this is 3'. So you will have a 5' here. I'll give some bases T and A. Okay. So here DNA polymerase should add. This is 5', and it should be adding towards, polymerizing towards the 3'. I'll just put this like this. So what base would it add? It will add thymine. What base should it add? It will add adenine. And here, what nucleotide should it add? Should it add actually? No. This is just an intercalating agent. It's not a part of the DNA. But then, because it is present and it can do some interactions, DNA polymerase may end up adding some base here. And then when it comes here, it will have to add thymine. So now you have an additional base added into it. And that is the problem. That is how additions can occur. And deletions can also occur if some of this could come on base pair while it is polymerizing. Okay? But I hope you get the picture of it. So mutations can be caused by agents like UV rays or X-rays, or it can be caused by base analogs or something like intercalating agents as well. So all the cells, if they have to remain as they are and should be, then they have to protect the genetic information. As I was telling, everything is information. Life is also information. If there is a change in the information, we wouldn't look the way we are now. We would look something else. I mean, it depends on how much change is happening. So there should be some mechanisms to keep the genetic information intact, unchanging. And if there is a change, I mean, as I was also telling, change is constant. And there is only one way of something to exist. One is, say, for example, this is an entity or genetic information. And we are saying that information will change. That means you have something to change like this. The only way to remain as we are as a species is to somehow change back to what we were, to repair the damage, to undo the changes. And that is what all of repair mechanism is dedicated to. If that repair mechanisms are not there, then we wouldn't have been having any kind of stable species. And we wouldn't have these many number of cells. I mean, these many types of organisms surviving. And that is why repair is something very important. It's the masterpiece of evolution, because that is what allows the existence of a species, continuance of a species. When I say species, here again, I would mean the genetic information. So there are several types of repair mechanisms. One of it is mismatch repair. Then you have direct reversal. Mismatch repair, we have already discussed that along with the replication errors. We will have a look at direct reversal, base excision repair. What does it say, direct reversal? Something, say thymidine dimers have formed, you can just make them as thymidines. That is directly reversal. You do not make big changes to the DNA. You don't cut anything. You just undo the damage. In some cases, you remove the base that is alkylated, for example, or oxidized. You have to remove those bases. And that is called as base excision. The repair starts with removing a base. That is what you should remember from the name. And in literature, you will find it abbreviated as BER, base excision repair. In the third one, in the fourth, it's called as NER, nucleotide excision repair. What is the difference between base excision repair and nucleotide excision repair? In this case, you are deleting a nucleotide, not just the nitrogenous base. Okay? And then we have something called as recombination repair, which I think we will revisit it when we are going to discuss about recombination. And then there is one that is called as translation DNA synthesis. That's my all-time favorite, actually. So this is the summary of all those mechanisms. And I think if you can, this is a good table to visit right before an exam, something like CAA or end semester exam. It's a good one to visit it because it summarizes everything and it tells us all the enzymes that are involved also. Okay, please try to go through it and what kind of damage the repair also is given. So that should help you a big time. So first thing we will look at is direct reversal, and which is also called as photoreactivation. So here is a DNA molecule, and because of UV light, you have the formation of thymidine dimer. And what is the ring that is called as? It is called as cyclobutane ring. It's a covalent bond between two dimers. There are multiple covalent bonds in there. So it is, if a thymidine dimer forms, the problems mostly are about base pairing. There is no base pairing, because of which transcription will be problematic and also replication will be troublesome. So there is an enzyme called as DNA photolyase. What does it say? DNA photolyase. That means it uses light and what does it do is it identifies the thymine dimers. To start with, it identifies thymine dimers, it binds there, and when there is visible light, okay, it uses that light energy to undo the cyclobutane ring. So it is direct reversal. Why is that? They are not doing anything at all. All it requires is just light energy, that is visible light energy, and then it is just breaking the bonds that make up the cyclobutane ring. And now we have the thymines as they are, and they can interact with the corresponding or complementary bases. Okay, it is called photoreactivation. The enzyme is called as DNA photolyase. There is one more direct reversal that is called as methyltransferase. The enzyme is methyltransferase. So some of the bases could get methylated, like in case of here, G-guanine is methylated. And that is a problem because it will complicate the stuff. We know the methylation of many bases. We know typically what we know are adenine is methylated, like by DAM methylase in GATC sequences. And in eukaryotes, for example, cytosine is methylated. They are kind of normal, or evolution has tolerated them, and they have a role to play in the existence and evolution of those respective organisms. But some of the methylations, such as in the case shown here, it is not an ordinary one. It causes a lot of problems for the DNA, so it should be removed. And we have something called as methyltransferase, which has a thio group, the SH, and it recognizes those methylated guanines, and it removes the methyl group. It is not doing anything at all except breaking the methyl group, and it forms a covalent bond with the enzyme, and it is removed. Now you have a guanine as such that it can base pair with the cytosine of the complementary strand. So the problem is it is very costly. Why is it costly? Because just because there is to remove one methyl group, now it has taken the methyl group and this whole enzyme should be degraded. Okay, there is no way of removing this methyl group just like that. The whole enzyme should be degraded. That means removal of just one methyl group, you are seeing that if, for example, this is made up of 100 amino acids, say, theoretically, the transcription of it and translation of a protein like this, and all it does is removal of one methyl group and it is dying it, right? It is very expensive to do that. For a bacterial cell, for example, to invest so much is a huge cost. And yet it is doing, which would only imply the importance of the genetic information, right? Everything, this is the genetic information is the base for every cell. If that genetic information is corrupt, then the cell dies. So even though it is so expensive, we still will use that so that we can survive and exist. That's how you should see why cell is investing so much. Well, you can always argue there could have been a smarter solution where you should have had some other enzyme which will just break off this methyl group. It is all possible. Evolution does work by logics, but it's not always the case. There is only one logic in evolution or in all of life. It is to, anyhow, I need to exist. That's the only logic. Any logic that concludes or results in or strengthens the existence of that organism, it will be there. It will survive. That's how you should see life as. So the other one is called as base excision repair. Okay, this is something important. I love this chapter because it gives the essence of everything that we are supposed to learn in molecular genetics, or you can take even the philosophical perspective, and it still has so many incredible things that are there in this particular chapter. I encourage you to read the textbook and watch several other YouTube videos if you have, and try to learn more about repair. Okay, here is a question. I have uracil in this place. There is a DNA, and the DNA has uracil. How might that uracil have come? It could be, I hope you remember, in spontaneous mutation, we said spontaneous deamination. Uracil and thymine are just different by one methyl group, right? Sorry, I put it wrong. Yes. N, and you have another N, and like this. This is uracil, and that's not part of, it's a part of RNA, and this is where, this is thymine. Right? Okay, I'll put here, that's my mistake. So if there was a demethylation of this, what would you get? You will get uracil. And you also had cytosine. In cytosine, what is the difference? One second. In cytosine, you would have had an amino group here, and this is the keto, that is cytosine. If you had deamination of amino group, that means the removal of amino group, which would have resulted in a keto group. Now you have uracil. So deamination of cytosine could result in the formation of uracil. That's the most predominantly why this might have happened, uracil here. So now uracil is abnormal in DNA and it should be removed. So what happens in this case is there is glycosylase. Uracil glycosylase, you would call it, because it is specific for removing uracil. Prasanna? My screen? Can someone say it again?
Yes, sir.
Am I audible?
Yes,
Yes,
sir.
sure.
Yes, sir.
Okay. Thank you. Okay, I think we are anyway in the last, so we will just continue with this one and then I will stop. So I'll give you an overview of this and then I will stop. And uracil is abnormal, so it should be removed. And the attack will happen on glycosidic bond between the nitrogenous base and the deoxyribose that is present here. And that is why we call these enzymes as glycosylase. It is breaking the glycosidic bond. And the first thing is they are removing the base, and that is why we call this also as base excision. Excision means cutting. You're starting the repair with base excision. And then there is, you have AP site here in this base. The first base that you're seeing, it has a deoxyribose, phosphate, and a base. And the last one deoxyribose, base, and a phosphate, deoxyribose, but no base. And that is called as AP site. It is for apyrimidic site or apurinic site. If you don't have an adenine or guanine, you would have called it as apyrimidic site. Now it is a pyrimidic site. That means it doesn't have the base here. And then there is an enzyme called as AP endonuclease. AP, as I said, AP, it is for apyrimidic or purinic site. It finds any of those bases and makes a cut here. And then there is an exonuclease activity. There is another enzyme, an exonuclease, will remove this whole base here. I mean, not the deoxyribose and the phosphate group. And now you have the takeover, and from this OH, it will start synthesizing new DNA. And that is how the gap is repaired. Okay? So I hope you understood this. In today's class, we have learned about Ames test. We learned about UV radiation. And I'll just quickly try to go back. Okay. This is also not. So I will just stop the presentation here. So we learned about nitrogenous bases, Ames test. We learned about base analogs like bromo, uridine, or so, which can get incorporated. And we also learned about intercalating agents, which could slip between the bases and cause insertions or deletions. Repair. In direct repair, there are two examples. The first example is for which reverses the thymine dimers that are formed because of UV radiation. And there are also other, there is also one more, that is the methyl transferase. Methyl transferase is an enzyme that removes methyl groups from bases. The enzyme, upon receiving or removing the methyl group onto itself, should be degraded in whole. So that's an expensive process. Then we were looking at the base excision repair. The title says it is removed. And then the sugar and we call the enzymes involved are called as glycosidic bond. Glycosidic bond that is present between the nitrogenous base and the deoxyribose. Okay? So we'll see in the next class. I hope some people at least are doing the second assignment. Please try to do so that I can be liberal enough. Okay?